this afternoon is me. So these are some key points that we'll be touching on. These are not all the key points, but these are like the main points that. So the main goal of this project is to increase the knowledge of the metabolic potential and influences in the five fungi, and to also um, also to learn about the cultural properties of the plant from the kingdom. So like I said before, the plant that we'll be focusing on is neem, which is used for its medicinal components found in the leaves and the stem, sometimes in the trunk as well, which we'll be touching on in this slide. So the specific aims that we have been touching on for the past 4 weeks is to identify and categorize the diversity of the fungal endophytes found in the two plants neem, which are other group will be touching on later, which is Malaya that our other group will be touching on later, but the main focus that we will be touching on is neem, otherwise known as a Draca indica. We've also worked alongside our peer mentors from the University of Nairobi. Okay, so neem, otherwise known as Asadrepta indica, is known as a miracle tree. Um, it's used, like I said before, it's used for its different medicinal properties found in the leaves and in the bark. Um, it's also used for its antibacterial, antifungal, and anti-clogging properties. Also used to relieve toothaches and other anti-allergenic pain. The main focus of our study are the endophytes, which are the bacteria that live in between the plant cells. Um, Endophyte fungi are highly diverse and are known to have secret secondary metabolites. So these are some of the accomplishments that we have successfully completed in the past 4 weeks. We did isolation of the fungal endophytes in the green plant. We subcultured the fungal mycelia. We also did some morphological characterization and DNA extraction. We did some HPTLC training that we'll touch on later, and we also did sample collection. So while here, we went to the herbarium and learned a few herbarium techniques. As you can see above, we learned to sterilize the equipment, sterilize the plant specimen. We also learned how to record the data, the information that we've learned on these plants, how to do background information, how to decipher the plants when we're out in the field. Um, and we also learned some of the techniques used to preserve these plants.
So, taking us through some of the steps. All right. Yeah. So, the fungal endophyte isolation began with the field the sample collection, as has been explained in the herbarium techniques, through the sterilization of materials saliently. This was done in Mara Machakos and also in Kilifi. After the samples were brought to the laboratory, there was the cutting and sterilization. Sterilization comprised some steps using 10% Clorox, 95% ethanol, and 70% ethanol. And then there was the cultivation of the prepared leaves and back parts, plant parts into previously prepared media. We used two types of media, that is potato dextrose agar and the malt extract agar. After the isolation, there was the preparation of the cultivation of the pure cultures of the attained fungi. And so from the neem samples, we attained a total of 8 fungi. And the first one was neem stem. From neem stem, we got 1 fungi. And then we had four endophytes from the bark, and we had 3 endophytes from the leaves. So the morphological traits, we described the endophytes using the morphology. What we could see, we would describe the plates using the upper part and the reverse part. Like, for example, these two, sample number 2 and number 3, we describe it as a black culture with a green reverse. After 7 days of culturing, it was 5.4 centimeters in diameter. And then sample number 3 was also described. It was bluish greenish on the upper part, and the reverse had some pinkish or orange color. The diameter was 4.3. After that, we did the microscopy for further identification. We were able to describe these as aspergillus based on the conidia form, the firelights, and the conidia. We were able to describe that sample as penicillium. And then there was the DNA extraction, it's still part of the description and characterization. So we begin by getting scrapping off the fungi from the plates, the pure culture we scrap into a sterile envelope. And after preparing the sample, we use a kit to extract the DNA. We extracted from, there were 8, as I said previously, the new samples were 8. And after running the agarose gel using a ladder, the standard of 1 kb, we were able to get DNA from all the samples. Yeah. So noting that initially we had a challenge in getting the DNA because when we were extracting, we just followed the kit and then we repeated and we used the manual crushing. That's how we were able to get the DNA.
So for our HPTLC results, the HPTLC results are supposed to include what kind of metabolites are found within the fungi, which are found within the samples themselves. So in order to obtain those HPTLC results, we started with cutting out the agar and the fungi from the plate itself, and then we placed it into a conical tube and added acetonitrile. We let it sit overnight and shaker for 15 hours. After we did that, we came back and we decanted what was in this tube, and we added within here DCM, which is dichloromethane, and distilled water as well. And we shook it for another 15 minutes, as well as included it into a centrifuge for about 30 minutes. And what you see here are 2 separate layers. We took the bottom layer of the solution and we used it to test for our HPTLC results. But before we even got to there, we had to rotary back those to basically break down the chemical to its natural state before we tested for HPTLC results. So here are the HPTLC results. Our ladder right here is a universal HPTLC analysis, basically. The universal ladder basically contains about 8 different chemical samples that are found within the universal ladder, basically. So from here, samples 1, 2, 3, 4, 5, 6, 7, and 8 are our neem samples. And we were only really successful with seeing bands from 1, 2, 3, and 5. And as you can see, when we align it with the universal sample, we can identify that some of the metabolites found within our fungal samples, which one in particular, sample 1, which came from derived from the stem, shows to be in alliance with 8, which is benzyl triol tetramico-butyl as well as sample 4, which is kind of unidentified. Further analysis needs to be done to really tone in on which metabolites are found. Um, we also see that in sample 5, that is also in alliance with number 1, but it's not really aligned with the universal pattern as well. Yeah, sample. Basically, 2 metabolites are found in sample 6, and 2 metabolites are found in sample 1. A lot of them kind of coexist. This one down here is just the base. This is where the sample was loaded. And the mobile phase kind of came up a little bit, but then it stopped. So we can hypothesize that it's somewhere between zero point 0.1, which is thymine, and guanosine, somewhere between those two samples. Further analysis needs to be done so we can really tone it on the exact. Oh, I'm sorry. Let me go back over it. So the mobile phase, the mobile phase will be used for moving those bands up. So our identify, we use hexane and ethyl acetate for our mobile phase, and for our derivatized, this is the derivatization picture, which is the final step of HPCLC. We use sulfuric acid and methanol for our derivatization specifically. So our field days, this is also about experiences as well, getting outside of Kenya and experiencing life. So these are some of our pictures of what we experienced while being here outside of the lab. A couple of pictures of our sample collection. So this is Manopi collecting some bark from the campus itself here in Chiromo. This is some of our pictures from Mombasa on the beach. A couple of Mombasa collecting samples from Mombasa, lead samples. Our visit to the giraffe park, giraffe center. Our visit to the national park of Nairobi. This is me and Tree collecting some samples, and this is our trip to Machakos to collect this further samples as well. So challenges, the biggest challenge that we had was HPCLC. It was our first time running it, and we had several trials so we can learn how to do it. We had a couple of mishaps as far as trying to really cultivate a good picture. So these were from our first 2 trials. As you can see, like the bottom layer, it was had a little accident. We dropped it into the mobile phase chamber, so it kind of smudged our results a little bit. So, as well as our derivatization, we also had issues with not allowing it to sit, our plates to sit in the mobile chamber for a long enough time for the bands to actually rise. So it took about 3 times for us to really perfect it. It still needs to be perfected, but yeah, that was one of our biggest challenges. So our future goals will be to collect more samples of neem and Melia from different locations, from different sites, including Arizona. I'm sure there will be more collections being held in Kenya as well. Further, another future goal will be PCR sequencing. So to basically specify and really tone in on the specific DNA of the fungi, so we can actually know the type of species. Like we know it's aspergillus and we know it's penicillin, but what species is it? So the PCR sequencing will help with that. As well as identifying the fungi species, and as well as comparing the metabolites from different regions, of which will include more HPCLC analysis to really tone in on, again, what chemicals are actually found within the samples of our fungi. And that's pretty much. Thank you so much. Questions? Questions from the floor?
That was a very good presentation from the 3 of you. It's hard work. Let's learn from them again. Now, I would like to ask you a few questions. Let me go back to your introduction. techniques. I can see you have referred to use of differences for identification and then the collections. So the herbarium techniques is supposed to be for identification. Now here, what do you think we should include to talk about identification? Is it when we talk about collection?
Well, could you repeat the question?
Yeah, and there are some, what do you use for identification? Is it
So,
the leaf size, the
yeah, so the point of the herbarium in general is to preserve our samples from what we get from the field. So that way we can reference them just in case that if we go back out to the field and that plant is no longer there. So the point of the herbarium is to preserve our samples. They also taught us how to give us techniques on how to collect the samples for preservation, to test for preservation. And this is what the techniques that they gave us about sterilizing our equipment, sterilizing our plant specimens, and recording where we got the sample from, geographical location. Um,
but nothing on the what to look for in
they,
the
oh, yes. They gave us a key. I'm sorry. They gave us a key when we went to go visit the librarian, which you should have included in here, but we didn't have like an electronic copy to place inside of our slides. But they did give us a key with like different characteristics of the specific type of plants that we're looking for. So that definitely helped us along when we were in the field I'm sorry. So yes, they did give us a key.
yes, so you should at least add a bullet there of the what to look for because you see during collection you do it so that you don't destroy those characters. Because if you destroy them, then you come with something the lab will not be able to identify. Yeah, so just another bullet to talk about the
What about
characters.
even using marijuana? So you're talking about the name. So what are the main characteristics that you
Yes.
look in? In this just from before we go to the field, the technique should help you know what to look for. So what are some of the main characteristics that you'll be looking for when you go to the field? I guess maybe they could have been captured here.
Yeah, that's what I'm saying. That's what I'm talking about. So maybe they could have another slide just to show maybe the leaf, a leaf of a leaf, those serrations,
Yeah.
leaf, yes. And then the fruit is also a characteristic. Is it the color or is it the size? You know? Yeah. Just another slide can cover all those characteristics to show some of the characters that we use. The number of petioles, the petioles of just one branch of the petiole.
But before we do this slide, please remember that this is the beginning. How about that?
Yeah.
So when you do the second presentation, it should we should have to improve it.
Yeah.
Well, I hope you take it from that point.
Yes.
Okay, so I'm wondering why they are different. This one has dash, dash, dash. This one has 1, 2, 3, 4, 5. And are there like different concepts you're following? I'm wondering on whether they're just bullets. So then why would we not just have the barium housing and that, and then this one's follow? Is it a slide presentation? Or that is what I'm asking. Why there are different types of presentation in the same? I don't know whether I'm making a point.
You are, but the reason why it was done this way, it could have been done a different way with the numbers being a lot smaller. The numbers were used to represent like the list form, so it makes it go in order for the herbarium techniques, which is why the herbarium technique is right above it. Now the bullet or the bullets on the side were topics that we wanted to make sure that we touched in on. That's why they're over on the side. Those are like the main topics of the slide. However, this is just a list to like what are the rundown. Yeah, just give you a slight little rundown on what we learned in the area.
Yeah, I think what she wants you to do is to have the title. It's okay to have this slide in 2 columns, but the title of the whole of it is a variant techniques. Then you have a subtitle for this and a subtitle for the other.
Okay. Thank
then it will clarify the issue. Nine, slide nine.
No, those are the first one also.
The eight also.
It's just the first one, the first, very first one. Yeah. So, you're doing name, and you have some beautiful plant pictures. Are they related to name in any way? The, the plants that you show me there, it's really nice. Would it be, I guess, I'm just thinking, how about if you had some name pictures?
Yeah. So the main reason why we have a little clip art of the plants around, we're just kind of thinking this way because our project is based on plants. So we wanted to just incorporate a little.
Well, name is the main one.
Yeah,
They're very beautiful. They're very beautiful, but we just want them to be the new parts.
Yes, ma'am.
Yeah, that's what I'm saying.
Parts of the
You already, everything about what you're presenting is name. So you capture the attention of people, then will be the first thing that will be there is name, picture. If you want to add others, okay. But definitely name must be captured. And I'm not sure that it is. Let's go to the second one. So the second picture would be name and like slide, like a professor has said. Yeah, before you even go to
This is the name tree. This is the
yeah, that's wonderful. That should have been like in your first one too. But it's okay. I know you are living
between
with
this and the other, they should just be proper new leaves like this, so that we see the yes. Then the stem also maybe at the back and the fruits.
and actually we put a lot of them in when we're in Mombasa. I don't know whether you have to get that, but I can share my own pictures so that you use your own pictures. But and I know she's leading the discussion, but I think that it would be nice to be a little bit chronological because from that one we go to isolation. But I'm wondering if we do skip something before we can do this sample isolation. Where do you get the samples to do the isolation?
The sample collection for all of the chocolates, which is the decision. Also this picture.
So what I think is critical, and even if you're reading papers, sample collection is a big deal. So it should have its own sample collection. How, what places you went and Ms. Brown has plenty of results on sample collection. It's not just because if we don't collect samples, we can't do all this beautiful work. So I know that I interrupted you, Professor, but I was thinking that before we even discuss, and this is for all of us, and if you didn't do it, it's fine. But we just hope that as you continue now to work on your presentation, that we can add of this. So I really would love a whole section of sample collection before you just make it. Imagine it's just a bullet, and at least I know that I spent 3 days in the field. Not how we were. And I don't know how many, I think you spend sometimes in Machakos too. So that's why it really is so important. We should have some section. But go on.
Here, I don't know why it cut off like that because it wasn't like that on my art PowerPoint.
Yeah, actually, that's where I'll come here.
Yeah, because I noticed that when it came on there, I was like, oh, they're gonna say something wrong, but it was not presented like that when we were making it, so it got lost in translation. This is a technical problem because I literally had it stem. This is the stem, bark, 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 bark.
Did you guys do this in Google Slides? Yeah. Maybe you transfer it to a PowerPoint?
But they
We should, yeah. It might have been transferred to a PowerPoint because sometimes it like messes up things. So just for like the future, like transfer it into a PowerPoint on your own and then look and then adjust it. When you need to. The thing is, I have that problem a lot. Yeah.
So she has answered the question before I asked her.
Because like when we go back to the other one, you passed another slide. So please go to the other one.
This one.
Yeah. And I think it's the explanation was excellent. But I guess for me and maybe there's some audience, people in the audience that are not very familiar with your technique, I would have wanted to know that you used, I know you talked about the media that you use, but I don't think it hurts to say, to include the type of media that is PDA, multi-extract, or whatever, at least in your, I guess it is a summary of your procedure. Yeah, I think we've done very well, but your cultures of cultivation, how about just mentioning what you and then of course we talked about sample collection, the organism section with a little bit more details. Where it is collected, something about the area.
Yeah, then on the slide, this is also a nice one. You show the front and the back. But it was also good to indicate how old the plants as well. Because like you see the one down there is using some metabolites. That's why the plant is brown.
That's about 60.
Yeah, yeah, but it's good to know at what stage this is when you're putting the metabolites. The pictures that was also very nice. You made a good photograph and a good, a very nice head. The head of this. Yes. And of course, you write it in italics. Just like fungal names. Yeah, this was also very good, a very clean DNA. And I'm also happy that you realized what your problem was, and now you macerated the wall well to be able to extract the DNA. Yeah. So different animal cells don't need a lot of maceration because they don't have a wall. They don't have a wall to the family, they require that maceration. And you know this step may not be in the kit, so that's why we talk about optimization of methods. Yeah, so then that one is not in the kit, but since they know that they always do that, we optimize that and add it to the project. So that was good. And what happened at work? This one.
It was just very faint. Like even this is our second charlatan. Even with our first charlatan, it was very faint. We followed the same procedure. I guess it's just that specific why it's just
It has DNA.
and it has DNA, but it's just
They probably needed a little bit more of maceration, or maybe the maceration was already too much, so you lost everything. Yeah, so in such a case, you now you should still the lab, you have to continue now doing the optimization until you get what is good for it. And then each time you also do such work, in science, you know, that is already also an observation that you note for that particular isolate. Yeah, then it arises questions in your head, why does it, this, that. Now, in this photograph, I was there. I'm wondering why I'm not appearing in the photo. Now, in this slide, I know you explained it a lot and very well, but I'm just wondering about this. Um, let's see. Yeah, yeah. Could it be that the mobile face also did not, was it too much?
So was this, I'm sorry, so what this depression is our derivatization. When we had used the machine to dip the thing into it, it didn't go all the way, it didn't touch the RF. So it only left the depression like this. So then we had to manually dip it in ourselves so that it can get all the way up there. So that's what this dip is. This is the derivatization. And it was the first dip too, so that's why it's so prominent.
So we can also say that this did not have a lot of time to move up and separate.
Maybe, yeah, maybe with the polarity of it, it didn't have enough time, but we did let it sit for like a good 30, 20 minutes in the mobile chamber so that the mobile base could reach the RF value. But this one is interesting. I don't understand why it's so bright. Yeah, I don't have all the answers for it. Everything, but it's definitely something to look into.
Uh, so, what, and you may have explained it. Please forgive me for asking again. All examples 001, 2, 3, 4. What do those mean?
001 are the samples, are name samples. So 001 is name stem, 001 is name bark, 003 is name bark as well, 005 is name bark. It's just our coding of how we coded our fungi and how we transferred it onto the pages.
Okay, so 001 is stem, or S for,
Well, it's just one, it goes 1, 2, 3, 4, 5, 6, 7, 8. I got cut off again because
but I'm looking at your label up there, S, D, 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 D.
yeah, and yes. So the S stands for stem.
Yeah.
The B stands for bark.
Okay.
But it is fungus from the back.
Yeah.
Yeah, I know the fungus on the back. So sometimes it really just helps if you have those codes in your presentation and then you didn't have time. But if you do, then you have a kind of a little legend that shows that 001 is something. Yeah. But my main question is, what was the ratio of your solvent? Because you just said hexane and ethyl, and so what is there? Was there a ratio?
Yeah.
Okay, what was the ratio?
70 mLs of hexane and 30 mLs of acetate.
Which in terms of ratio is more than
So, what?
yeah, this is 100%. So the 70 is to 30. Yeah. Well, it's just nice to have this. Yeah. And I guess we didn't have a lot of time, and there was very good explanation of everything. But just a little summary on the slide will help people who probably want to code this and probably have just that's I think my general comment that it does help to have some explanation on the slide, even though you can talk about it because it's presenting. It's interesting to see that you have a band. The bands are not very well defined, but you did explain that maybe it was a little challenging. So I guess as is perfected, it will be maybe more clear the bands.
It was better than our first 2 attempts. This was the best attempt.
Now I see why I kept on asking and you share. Okay. very good attempt, and practice makes perfect. So
These were our first two attempts. We're still trying to get the hang of it.
Yeah, I like this slide because it tells me that you're still working and that you have goals. Isn't it?
Yes.
Yes. And that you let me know if you have achieved your goals. And I like this also because you learn some useful words. Yes. So, any questions? Right.
you only may go to make the neck into better. So we are not criticizing. That's always my first statement. But we just want to make it better at the end of the day. When it is better, we are all welcome. So we pray that we hope that you take any word that we say positively. Please use whatever comments to just make it better.
It's good. And at least when you had the presentation, you realize that you didn't waste 3 weeks, that you actually worked. Yeah. Yeah. And also, when you make your presentations, remember to put in all your findings. Because, like, for example, the characteristics for identification, if you leave it out, then it reduces your output. Somebody will not know that you learned this. So whatever you don't put here, it's not counted. Yes. Whenever you make your presentation, make sure you put all your results available.
Your acknowledgement page should have a list of people. Yeah, it's okay to get pictures, but please have an acknowledgement henceforth with a list of people that you are saying thank you to.
You don't exclude this.
Yeah, this is
That's
this
the
is
next
great.
one.
Yeah, this is very
Another
nice.
slide. This is very nice. Because it shows you actually love. It shows that you are in the field and you identify the largest tree among the others. So just the next slide of acknowledgement. All right, let's clap.
So what do you think first?
Yes.
But everyone, you are the ones who are first, so the others are now okay.
Yes. Oh my goodness.
And I also like the way they distributed their presentation according to their expertise. I noticed that.
Yeah.
Somebody was very good at collections, somebody was very good at HPTLC, she was good at the job, somebody does the counters every day, was told to do the counters.
Yeah.
So you identify your strengths, then you share your presentation. All right, let's welcome the next slide.
Good afternoon, everybody. My name is Vanessa Cherry,
I'm Dahlia Brown.
And I'm Chukwusa Makobe.
and the title of our project is Tropical Plants, Their Ecosystem Bias and Metabolism. Our PIs are Dr. Endosano, Dr. Jing Ping, I'm sorry. Our senior scientist is Dr. Betsy Otto. Our collaborator is Dr. Shona, and our few mentors are Chikusa, Amakope, and Louise.
So we're going to start with some background on our plant that we're focusing on. Our presentation is on Melia. Melia is actually native to Southeast Asia and parts of Australia, but it can be found all over the globe. It is colloquially known as Melia, and it is used in many different medicinal remedies, including treating skin conditions, and snake bites. And for many years, Kenyans have passed down the knowledge to use these plants to for their medicinal properties and using the metabolites, which are thought to be produced by the endophytes in the plant. Oh, and that's a picture of one of our collections that we did with Amelia, and I wanted to include this one because it had the flowers on it, and Dr. Osano said it's kind of rare to find those. Next slide. This is going to be some background on endophytes. The word endophyte is made up of two words, being endo, which means within, and then phyton, which means plant, so it's within the plant. An endophyte is described as an organism, fungal or bacterial, that forms a symbiotic relationship with a plant, but that symbiotic relationship does not always have to be mutualistic. It can be pathogenic, and but endophytes can also facilitate the uptake of additional water and other nutrients. So it's kind of, it can be good or it can be really, really bad. Next slide. And then some factors influencing endophytic population is host specificity and environmental conditions. So the endophyte will want its host plant to survive. So if it's in a nice environment that normally gets water, but then out of nowhere there's a drought, the endophyte will work towards helping the plant survive with like less water because it's trying to worry about itself.
So the goal of our project is to increase knowledge on the metabolic potential and influences of endophytic fungi in culturally important plants in Kenya. And our specific aim is to categorize the diversity of fungal endophytes in leaves of two plants, which are as a direct indica, but our project is specifically focusing on Media azerua, with surveys and collection of plants born in Kenya and in the USA with student participants from Bombay State University as well as the University of Nairobi. Our accomplishments for the first 4 weeks of the project here in Kenya included sorting media, isolating fungal endophytes from media leaves, subculturing fungal colonies, and once we had the pure colonies, we moved on to morphological characterization, which was followed by molecular characterization, which involved DNA extraction of fungal endophytes and HPLC training, and the extraction of the metabolites produced by those fungal endophytes. And that was followed by additional sample collection from Machakos, Kilifi, Kwale, as well as our very own Chiromo campus. Next, we came back to the lab and isolated those fungal endophytes from those plants and prepared them and preserved them as well in those plants to be transported to Arizona. And also, I don't want to forget to mention that we also did herbarium collection training where we learned different techniques for collection and preservation as well. For materials and methods, we started with sample collection. We collected samples from 4 sites, as mentioned earlier, which were Machakos, University of Nairobi, Kilifi, Kwale. And in total, we had 4 different counties, and in those counties, we had 6 sites and 21 streams. And here is a map of the sample collection with a description of the different climates. Here in Nairobi Chiromo campus, we had 1 site and collected 3 million trees. And that very from Nairobi is Machakos County, where we also had 1 site with 3 trees, and both of the sites, the climate were semi-arid. And if we go over here on the coast, we went to Mombasa, 2 counties each. And between those 2 counties, we had a total of 5 sites and 50 trees. And the climate conditions for those sites were tropical slash semi-arid, depending on where you went in the area. The material we used to make sample collection were ethanol in different concentrations, petri dishes, plates. We used both MEA as well as BZA plates. Of course, we had the cutting tools for cutting the leaves from the tree, paper bags and Ziploc bags for storage, as well as 10% bleach the surface sterilization, which we'll get into in the coming slides. The sample collection procedure was as follows. So first, when we get to the tree, we started by documenting relevant information such as the geographical location, climatic conditions, the height of the tree, its age, the weather conditions, and such. Next, we use aseptic techniques to collect the samples from each tree, store them in paper bags, and took them back to the lab where we moved on to the next stage, which was fungal isolation. For fungal isolation, we started by surface sterilization because we wanted to eliminate contaminants as well as endophytes because our goal was to isolate endophytes. So what we did was we used 95% ethanol, 30% cloth, as well as 70% ethanol to eliminate those unwanted microbes, and then moved on to transferring those segments of the leaf onto plates, and that was followed by incubation, and we allowed it to grow. The next step was self-culturing because once we would remove the plates from the incubator after about 7 days, we would observe the growth of multiple different colonies. So we wanted to cultivate few cultures of these colonies so that we could conduct further characterization of those. So we subculture them by preparing fresh media plates, and we used both PDA and MDA plates. We transferred each colony to spread the mycelia from each colony into a liquid of its own, labeled it, and this is incubation into the incubator for growth. So once we had our pure colonies, we moved on to morphological characterization, which is where we would describe the fungal colonies appearance by looking at the color of the front and reverse of the plate, measure the diameter, describe the media we use, its age, fruiting bodies, as well as any exudates produced. And here is an example of a simple MLA, which stands for Milia A. And if you look here at the front, the color in the center is light green with a brown exterior, and in the back, it is completely dark brown. And the diameter was 55 millimeters, the media was 58, and the age at the time we did the observation was 7 days. Next slide, please. This is another example of the sample, Melia B. The front was white and fluffy, the back was also white. The diameter was 60 millimeters in length. Media was PDA, and it was also 7 days old. The next step in morphological characterization was actually to conduct microscopic characterization, where we would press slides and look at them under the microscope to try and identify and describe the hyphae, the chlamydia, its shape, its color, whether it's smooth, rough, and the arrangement, as well as the structure of the fruiting bodies. And here is an example of one of the microscopic characterization. Here we identify the chlamydia, the vesicle, the chlamydia core. Next.
So we come to the DNA extraction, and it's important to note that if we compare the mean and the median, we isolated fewer colonies in the median as compared to the mean. And in the media plant, we had the media samples for the leaves and for the bark. And we did not isolate anything in the bark, but we isolated in the leaves. For the DNA extraction, we used the Zymo kit, and we, as Madison said, we modified the protocol, and we were able to get DNA. And later on, we did gel electrophoresis, where we prepared 1% agarose gel containing 4UL, ethidium bromide, and we ran it using the 80 volt and we viewed it. And then later on, we were able to do the HPT assay.
So this is an image of when we were crushing it and then centrifuging it, and then just the whole process for DNA extraction. And then we can move to the next slide. So this is the image that we obtained from the gel electrophoresis, which is you can see the ladder at both ends, and it can be able to you can be able to estimate the the, the size of the DNA that you obtained. So for the HPTLC, for the preparation, step one, we did the harvesting of the mycelial plants from the plates, and then we put it, we transferred it into 50 ml Falcon tubes, and we added 5 ml of acetonitrile and let it sit for 15 hours. The next day, we decanted the liquid and we added water and dichloromethane, which was later followed by shaking for 30 minutes in an orbital shaker. And we centrifuged it at 3000 RPM for 15 minutes. And where we took now the bottom phase and we took it to the rotor evaporator, and now we were able to get our sample. So basically, the HPTLC protocol, first of all, you load your sample in the Linomat 5. Oh, okay, before that, you were to take your plate, you check whether it's good, no scratches, and it's good. So we load the samples in the Linomat 5, we visualize it, and then we take it now to the developing tank for 30 around 30 minutes where we were using hexane 70 and ethyl acetate 30. Then we come back to the visualizer again. And then we do the derivatization step. After that, we heat and come and visualize again. So we were using the program, the Visual Cards. So this was the results that we got. And our sample starts from here, sample 009. So as I said earlier, from the mania we did not, were not able to isolate many colonies. So from this is mania leaf, all of them 9 to 12 mania leaf. And you can compare it to the name plant. You can see only sample 9 was had a band. And when we use the UHM standard, we can be able to inference that sample 9 had this thymidine, thymidine, thymidine compound.
Our objectives for the next 4 weeks are isolating the samples while continuing to isolate the samples that we collected in Mombasa, which or the counties of Kilifi and Kwale. We're also working on identifying the fungal endophyte type for said samples using the morphological characteristics and using the keys that were given to us. Um, we're also going to be running HPTLC on the fungal endophytes to identify which chemicals are present and compare the endophytes found in Malia across the regions and species, as we were lucky enough to find another species of Malia on our travels. And like everything, there were a couple of challenges. Next slide, please. Our first challenge that we wanted to touch on was our first HPTLC did not produce clear images like the pictures that were shown in the other group slide. There were not clear bands we couldn't see. We addressed it by repeating the procedure and going through it thoroughly, and it produced some results. Our second challenge was our first gel electrophoresis did not produce results for many of the samples. Our strategy was to just rerun it again, and we modified the procedure. And that's when the slide that we had earlier in the presentation that had all the bands, that's when we got that one. And then for challenge number 3, most of our first endophyte isolation plates were contaminated. That was our fault. We poured the media outside of the um, back then. So we evaluated what could have contributed to it and decided to pour the media in the biosafety cabinet in order to reduce contamination. And these are references from the PowerPoint. In our acknowledgments, our gratitude is extended to the NSF for funding this research project, our PI, Dr. Ann Osano, our co-PI, Dr. John Nam Kang, our senior scientist, Dr. Elizabeth Betsy, our collaborator and host, Professor Sheila Okal, and our peer mentors, Rosa and Luis. And yeah, and that's us in the lab. And next slide. Sorry.
Wow. Thank you very much for your beautiful presentation. You wrote everything that you had and it should not be made. Nothing reminding you computers. Any questions from the audience? Any question or acclamation or encouragement?
I really like it was very yeah. I like how you guys went to detail. I feel like we kind of simplified it, but you guys went to detail, which is a good thing. In this situation, we were like, well, more is more. So we're just gonna pay everything, and hopefully, if you guys are like, no, this is a little too much, like tone down.
Place everything there. All right. So let's start with the last slide. Now this one. Tell me, you want media outside cabinet?
Yes. Because sometimes like normally at home we kind of if we don't have enough.
Where was Tuposa?
They were not supervising us. They were in the other room. So we were pouring and yeah. I think what the thing was like we thought we didn't have enough room in the bio cabinet to really have all the plates inside there. So we were like, maybe we should just pour it outside. Yeah. To maximize space. Yeah. We just sterilized the surface, but we thought that the contaminants came from the air.
From the air.
Yeah.
It can't come from the surface.
Yeah.
It comes from the air. Yeah, but that's a good experience because it shows you what's in the air. Yeah. And it also shows you what happens to your body. You don't get sick because your body is always fighting those things in the air because you breathe them in. Okay. Yes, that was a good experience. Now, let's go now to slide 1. So this counts. Yeah, but it's a good thing to just write everything and then. Yeah, but if you go for a competition, it's good to write everything, yes, as you find first slide. Then after you've done this and sat down in your room, you have to go back to the call and find out how many minutes, how much time you're given for your presentation. Then you can practice and see how long this will take. Then you come back now to mold it now and make it small. And now let's look through and see where you can do that. Now this slide, can anyone tell me what can be done with this one?
That's some bullets. Bulletin.
Yes, bulletin. I thought it was it's nice. Yeah, it's nice, but the fonts are too small. Because you see, you have a lot of space, and then we struggle to read. Of course, the font is beautiful and it's a good size. Yeah, but there's still a lot of space here to have less trouble in reading. The font is small. And I think you change. Because whenever you choose a slideshow, a slide type, if you don't change You change, isn't it? Yeah. If you don't change, then it gives you the right font. Yeah, so this one you manipulated, and it became now smaller fonts. So in most times it's good to stick to the font that that slide gives you. Yeah, because it controls now the amount of information you put for the audience. It helps you. Of course, you can manipulate a little because for scientific things you have to put a picture, but have it in mind that it helps you control the content of the slide. So if you manipulate, then have the conscience of not putting too many things to change the font. Otherwise, the photograph is very beautiful with the flowers that Susanna said is the key to identification. It looks nice.
And it is so
To get
flowers. So, I've gone to field trips. I don't know how many times I've seen the flowers.
Yeah, okay, so it's good to know this season.
Yeah, that was really
Yeah, it's
a
good to know
wonderful
this
season.
season.
And
Yeah.
also the fact that for the 4 of you were in the field,
Yeah,
even the area, because we found this in Kwale. Also, yes, it wasn't in
so you know the area, you know the and even the rainfall, the temperature of that season. Yeah, because those are the things that stimulate plants to flower.
Yeah. And that's why and I think you did a good job with the collection. That's why I was talking about collection. Because we'll be talking about the site, what was the climate, what those elevations and everything. And so if somebody is looking for these flowers, maybe the flowers do something. They know that I can go to this location, this time. If you're coming from the US, don't come in January, come in June, whatever. Yeah. So little things that is so important. Yeah.
Then if you have this name here, then I would think it is the name of the plant.
Oh no, yeah. I just was so used to that. In my other PowerPoints that I normally do, they always ask you to
It should be the name of the plant.
what's the name of it?
Yeah.
Yeah.
And yeah, like a little description of
source.
how they many are flowering, because it's such a great, important something for people to notice.
This is your name?
Yes.
Yeah, I just say, I said fine, but you just say collected by.
Yeah, I was just used to all the time they
No
say
problem, just say Delia Plant collected by Delia Brown. And here I am. It's like pretty. Yeah, then slowly you will also learn to make slides by not writing stories. Yeah, making them a little shorter. Like an endophyte is described as an organism. For slides now, you just write endophyte is an organism. We reduce the, those wastage words. It's only on slides that will allow you to make a complete sentence. Yeah.
Just like we go see, I think that's what Professor is saying. Enterprise can be individualistic or pathogenic. It's not a complete sentence, but the message is very clear.
Yeah. Yeah. So there's a lot here that you can reduce. Even the first sentence, the word endophyte is made up of two words. These two words being endo meaning within and phyton meaning plant. So in a slide, you just write endophyte, then you can enter and then write endo is equal to, and then uh, phyte, phyton equals to plant. Just equals like this. Yeah, you don't have to write it as a sentence. Then it reduces the busyness of the slide. So you can learn that slowly in all your slides. It's something that is in all your slides. Slide 6. It's like now this one also is a very long sentence. So when you start reading it, by the time you reach base, you are asleep. So maybe you can
I think I gave them that and they just put it like this. Yeah.
or they cut and paste.
Yeah. But you see now, because you're just looking at as a delay, for example, you didn't have to write as a director there.
Yeah.
Yeah. So that's just ways of reducing what you're saying.
Yeah, because the presentation is yours, so just write yours. In fact, first I was even presenting for me. And when she was presenting the results, she was also presenting the new plans.
And then, yeah, that reminds me of the first slide. Is it possible to quickly go back there? I think I saw it, but I forgot. So, no, first one. That's where your title is, and there's no mention of your plant. Yeah. So I think this group at least say semicolon name. So it has to, your plant must be there.
Yes.
I think we have to find a way of this tropical plants and maybe name for that matter. This was a big topic for the NSA.
Yeah.
So we can tailor make it to fit our own specific study.
Yeah. So this is the topic of the whole project. Yeah, but you're talking only about me. I've got my letter. Yeah, so that will help reduce that one too. What do you want to say about this?
I like the way you've done a lot, and you captured everything. So I really like that. Um, I was just thinking that after summarizing, and I was so excited, so I want to see how each part.
And also maybe they add 2023 because this would be
Yeah.
1994.
Okay.
Before they were born.
Yeah. Um,
I like this also. You were so detailed and nice. But now when you are here, you read this. I don't think Kenya looks like this. I know you wanted to show me where Machakos is and Nairobi. But you pulled it up. What is happening?
When you're making drink, you do the corners. You don't do like this. You do corners and then it's proportional. Yeah. But go back to the other one which you liked, which I also like. This is very nice. And I'm thinking that if there's a way that you can use this as a sample for the rest of your slide, it's very well done.
Yeah.
This font is just right.
Yeah, this one they didn't manipulate.
Yeah. So something like this. So what is that?
Don't worry.
I'm worried. I don't know what I'm supposed to have.
This was also nice. But when you talk about bags, someone will not know what Ziploc is. Ziploc and also paper. That is because it is just the zip that is locked. Yeah. So maybe polypaid or something. Properly. I like those chocolates. Yeah, but you could reduce the writings so that they're not too busy. Because the fonts now becomes small. It is a good way of presentation and then summarizing. Yeah, I like this also because you showed what you are trying to talk about the plant also. what were the characteristics that you are looking for in a fungus. Yeah, the characteristics that you use for. Yeah.
Where were they in the plant? This nice characteristic.
They are now in the plant now, they didn't show.
I know. We are just being reminded now. You did a fantastic job with the what cotton is. It's so nice to have something like this for the plant.
Yes. Yeah. Yeah, because this is nice. You can put the extrudates, which is very good because it's one of the characteristics we use in an education of fun. The fruiting bodies, whether they have or not. This one. Maybe that's why you took that night. Now I see it works. Oh. And then it's yeah. Keep trusting. Yeah, there's something else I wanted to talk about here. Is that when you start doing your work, I know you like just scrapping quickly. Yeah, but you see, you just notice that I should photograph this vegetation. But it finds when you have just labeled the things, so you can't even use it for publication. So normally I encourage that whenever you just start doing your isolations, just label in a nice way. So that in case by God's grace a miracle has happened in your petri dish, it's still presented for publication, it's still in it. If you want to use this to publish, for example, you now struggle to start wrapping off those parts. Okay, so just have the habit of labeling your petri dishes in a smart way so that you don't have to wrap, to block, to be photographed.
But is this an example? Why is one example there?
It was just an example of what we were doing. We've done it with a bunch of different plates. We just wanted to show you guys what we were actually looking at to kind of try it.
It's an example of using this key.
Yes.
Color, diameter, media, age. So here now she's telling you that it is 7 days old, the age, the front is this, the diameter is this.
Okay. I hear that, but would it just be enough to? Is this part of? But this is part of the research.
Yeah, it's part of the education, but now they look down already hour. So she doesn't know the name of the but she has the characteristics in stock, waiting for the
How about just saying molecular composition result? I don't know. And this tells me that there are more. So did you have actually? Let me just ask my question then. Did you have more cultures?
Yeah,
So that those are two.
Yeah,
So
we only had 4.
Oh, you only had 4.
Yeah,
Oh,
just
yes.
4.
Okay, so then there were just 4.
Yeah, there's
Okay. So they're not examples, it's just that this one,
mm-hmm.
the 4 cultures we isolated. That to me is a story.
Yeah, it's
So
just
when you say it's example, I'm thinking there are $100.
Oh, so
And so I was looking for these $100 that where are they?
So just write instead of example 1, isolate 1.
Yeah, isolate 1.
Yeah, then isolate 2. But since there are 4, you should have just photographed the 4 of them.
And put them in one slide or something. Maybe because you are describing.
Just have 4 slides.
Yeah.
Each one of the slide.
Yeah, they are very pure concepts.
They are very nice. These are very nice. And this is actually an endophyte. This white one. They like growing like that.
Oh.
And refusing to sporulate.
Oh, they don't sporulate.
They are very difficult.
In fact, one time we went 3 months.
Yeah.
No, it took 3 months to grow, not even to sporulate.
Yeah.
Until we were throwing dishes, then we saw it started growing.
Yeah. You know, in the plant inside there, there's not much growth, so they've evolved to stay there, that each place.
Yeah.
So when you put them in the plate, they have to remember that they are now have space. Then, this one also, I like this also.
I really think it should be the picture on my report
Yeah.
for when you pass.
This girl took my book and
This
she
is
discovered that
good.
she her name Lydia. And
Yeah, this is whoever did the picture, very well done.
yes,
Very well done. And of course, the lady.
yes, I'm Lydia. That is very nice.
Oh, that's the one you didn't even worry about.
Okay, now this one looks like the other one I saw there.
Yeah.
Did you steal it or did you,
No, we
what happened?
It's the same one.
Yeah.
The one with all.
we use the same.
You shared.
Yeah.
Okay, so earlier in the project, was split into two groups, and I think it was me and we working together, so we had an image, and Dahlia and Madison, they we had an image. So this, what Dahlia used was the one that she Madison shared. And Madison also used that one.
So you find out in there.
Yeah.
Yeah.
Just an example of one of the results we had.
Yeah, actually, my reason of just bringing it up is that sometimes in meeting we can take somebody's picture and put. Then one of your participants remembers that they saw it somewhere. Yeah, yeah. So if it's used, then it's fine. Yeah. Then this now is the last one. Sorry, I have too many questions. Yeah, you see, this story is a lot. It's like an essay. It even has a full stop. Then the sentence continues. Three full stops. Yeah, so you learn to put bullets and also not write essays. So this is one slide that can be reduced. Okay. Oh, they are the monkeys.
Yeah, that's what I was thinking. I was thinking of the monkeys running.
So anyone else with a question so that we end this?
No, it is
Anyone with a question?
it's ending. So I don't have a question.
All right, so let's clap for them. Okay. So you just use the comments to improve your
Yes,
application.
yeah.